Hello, everyone. Thank you for attending today's webinar, De Novo Design of Allosterically Switchable Protein Complexes, presented by Baker Labs in partnership with Refine. I'm Rebecca Kushmiter, and I'll be moderating this webinar. All right, now I'd like to introduce our speaker. We'll be joined today by Arvind Pillai, postdoctoral fellow, Institute of Protein Design at Baker Lab. You can read his full bio in the speaker's bios panel. All right, and with that, I'll hand it on over. Arvind, please go ahead.
Hello, my name is Arvind. I'm a postdoc in the Baker Lab. Thanks for coming to this webinar. And I'm going to be talking about some research we've been doing on the de novo design of dynamic assemblies or allosterically switchable protein complexes, particularly assisted by mass photometry techniques. So starting off with some basics here, most biological functions or biochemical functions in the cell rely on the activity of proteins. So in order to function, most proteins have to do three things. First, they have to fold into precise three-dimensional protein structures from these extended polypeptide chains made up of many amino acids. And that's a complicated folding process. Most proteins don't function as isolated molecular units. They assemble into higher order architectures of protein complexes held together by non-covalent protein interactions. And finally, proteins are not static objects. They can toggle between alternative conformations driven by binding events. In this case, calmodulin responds to binding to calcium by undergoing this large rigid body conformational change. And that's pretty central for lots of different kinds of signaling and regulation in the cell. And so in this talk, I'm mostly going to be discussing how we can design from scratch these latter two features using deep learning-based computational methods. So virtually every process in the cell relies on the ability of proteins to respond to signals by altering their structure and function. And by turning proteins on or off, cells can impose control over their signaling or their metabolic processes. And that's symbolized here, where you can envision like there's multiple enzymes that carry out some biochemical process, and you can turn them on or off selectively by adding in appropriate regulatory molecules. So the way this is typically accomplished is through this biophysical phenomena called allostery. So allostery is a phenomena where a regulatory molecule, usually called an effector, binds to one surface on a protein and induces some change in its structure that results in a change in its activity. And the effector binding site and the active site of the protein, where, for example, it's an enzyme that carries out some reaction, could be separated by tens of angstroms. So this makes it quite interesting from a biophysical perspective because it involves action at a distance. So how is it possible that something binds to one side of a protein and communicates a signal through a protein to a totally different site? And a huge amount of literature is focused on kind of why or how proteins accomplish this complicated molecular task. So historically, by historically I mean over the last like 60 years, one way that biochemists or biophysicists have tried to understand this phenomena is by taking proteins and kind of mentally imagining them as simple machines, or analogizing them to simple machines like levers, gears, and springs. So these are systems with just a few different states and like limited degrees of motion, and they undergo these big rigid body changes as they carry out their functions, and that ultimately allows them to respond to signals. But as time has passed and our biophysical and structural understanding of allostery has grown, we now know that protein allostery is quite a bit more complicated than like a pulley or something like that. Some of the changes that are induced by binding to a protein structure are actually quite subtle, and many residues are involved and many states are involved. And that kind of obscures the biophysical basis or genetic basis of these features, like what residues are actually involved and what the mechanisms are. And also it imposes a huge limitation in terms of redesigning these systems for new purposes, which is something we want to do as synthetic biologists. So kind of the premise of my postdoc has been to what if it's possible to build new sets of proteins, not the ones that evolution has generated over millions of years, but new kinds of proteins which are rigid and robust, and that can actually capture the simpler mechanical vision of allostery, and maybe that's more rationally tunable or modulatable, and can be deployed for new tasks that never evolved in evolutionary history. So it's worth saying here, though, why would we actually want to have allosteric proteins or design them? And there's actually a wide variety of different reasons. So basically, we want to be able to synthetically reproduce some of the things that natural proteins have evolved to do to solve different kinds of tasks. One example of this that's quite relevant for sensing and processing signals is cooperativity. So cooperativity is arguably the oldest type of allostery that scientists have studied over the last 50 years. So it's a special case of allostery where if you envision proteins assembling into a complex, the binding of a regulatory molecule or a ligand to one protein in the complex induces a conformational change that increases the probability that the next subunit in the complex can bind. So what this allows for is for proteins to assemble into these multimeric, like multi-subunit architectures that undergo these concerted or coordinated molecular motions. And that's important. This all or nothing response is important because what it allows is sensitivity to a signal over a narrow range of ligand concentrations. So basically you have this switch-like response that's rapid as like something that you're trying to sense rises above a threshold. So you want this like kind of S-shaped curve shown over here. That's the signature of cooperativity. And one classic example of this, and I'll give another one soon after this, is like ion channels that conditionally open up. They're embedded in a membrane. They sequester some ion, like calcium, for example, but they don't open up unless there's a sufficient threshold concentration of this green ligand shown here. And once it does, it opens up and you release calcium and that generates a signal. And that's like a hugely important thing in all kinds of cell signaling. It's also important for, from a therapeutic perspective, for triggerable delivery, where you have these nano cages that can sequester some kind of drug, and when they encounter their appropriate environment, maybe it's a tumor microenvironment or something, they sense something that's unique to that environment, and then the entire cage disassembles and unleashes its payload and hopefully kills cells in a targeted way. Another important thing that's sort of related to the cooperativity question on the left is biosensor design. So we sometimes want to sense biomarkers for different diseases, or these could be small molecules or peptides, bind them with some sort of protein that undergoes a conformational change and then switches on, and now it can process some substrate over here shown in black into a product that is readable using some kind of plate reader device. So it's a luminescent or fluorescent signal that can be readily registered at like a macroscopic level. So, but using that, you can like detect hopefully important diagnostic molecules with high sensitivity and low noise. And finally, like the grand design goal of all of this, especially in the Baker lab, is to ultimately build molecular motors, so systems that can take some chemical fuel and convert it to mechanical energy. We're still, kind of inching slowly towards this ultimate goal. Maybe solving the first three is getting partway to there. But the idea would be that we can kind of replicate some of the walkers and rotors and pumps that you find in nature that can do all kinds of useful tasks. So to start with, I'll talk about the first of these. How do you actually design cooperativity? It's actually a very subtle phenomena, and we were basically able to use mass spectrometry to actually really interrogate and quantify this property in completely synthetic systems. And I'll also talk about the other two of these after this. But yeah, so I'll start with the most canonical example of cooperativity in all of protein biochemistry, which is hemoglobin. So it's a heteromeric, so a 4-subunit protein complex that's responsible for binding to oxygen in the lungs and releasing it in the tissues. So to do this, it has to have a high affinity for oxygen in a high oxygen environment and a lower affinity for oxygen, a low oxygen environment. And this is made possible through cooperativity. So it's this, again, once again, this S-shaped curve allows it to efficiently transport oxygen between these two different environments. And that's ultimately made possible by the dynamic motions of the complex. So it's able to sample these two different states I'm showing here by going between these large rigid body motion between two different conformations. So people have been studying this for a long time, and one of the first quantitative models for how allostery works was called the Monod-Wyman-Chenhua model. And what this proposes is that there's ultimately the molecular basis for this offloading and onloading of oxygen is encoded by the ability to switch between two different states, two different oligomeric or multimeric states, high assembly states, shown in X and Y state here, that are both symmetrical and that differ in their affinity for, in this case, oxygen. And if you are able to set up a system like this that can selectively go between these two states, you can deliver the kinds of cooperativity I'm showing on the left here. And in this case, by the way, the red thing could be an arbitrary ligand. It doesn't have to be oxygen. And in fact, in the subsequent slides, I've mostly explored cooperative binding to peptides, basically. All right, so there are two tasks ahead of us. So to construct something that can actually have two subunits to somehow talk to each other and influence one another's affinity, we have to go through two steps. The first step is to generate something that can bind to a ligand and then undergo a change in shape. And by the way, we're trying to do this from scratch. We're not taking anything in nature and trying to just modulate or modify it for a new task. We're trying to build this from the ground up. So the first step is to make a confidently switchable protein. By itself, this protein would not have any cooperativity, right? It would have this hockey stick-like hyperbolic curve, which is canonical for a non-cooperative binder. So it wouldn't have that S-shaped curve. But the hypothesis we have is that if we can integrate this into a larger complex that can sample just these two different symmetric states, we will have cooperativity on our hands as like an emergent phenomena, if the MWC model is true from 50 years ago. So before I get into like the two-state design aspect of this, maybe I'll briefly discuss like what is protein design, how does it work in a little bit of detail. So there's basically three steps to protein design. And I should say also that over the last maybe 3 years, there's been a huge sea change in the methods used for designing proteins. They've retained this three-step process, but it's been now informed with neural networks that are good at understanding and designing and predicting protein sequence and structure and function. So the first step is to build like a protein backbone. You can think of it as like an empty scaffold of carbons and nitrogens that onto which you can then decorate a sequence in a second step. And so in this step, you basically generate it by either using a diffusion-based generative AI approach, which can take a cloud of atoms and then denoise it to eventually get to a physically reasonable backbone structure, or you can do it parametrically, which is how you used to do it previously, where you just exploit your knowledge of the physics of torsion angles and the parameters that describe helices to actually suture together a protein backbone from scratch. So the second step then is to then take your scaffold and then identify a sequence that can be threaded onto it. And this is usually done by a second neural network. In this case, it's protein MPNN, protein message passing neural networks that can take in a protein fold and then give the most optimal sequence to satisfy that fold, or give several sequences that could satisfy that fold. And then the final step is to ask another neural net developed by Google DeepMind called AlphaFold 2, which can take in a protein sequence that you've decoded from your fold using MPNN and then refold it to see if it actually folds to the expected target design. And we can quantify that just by taking the outputs of AlphaFold when we feed it in a sequence and comparing it to our original backbone that we wanted to design. So obviously this process involves some errors and filtering. So by the time you get past the AlphaFold step, you may have filtered out a vast majority of like the sequences that and backbones that you initially designed. So the next step is to take that and then feed that into a pipeline of experiments. So you can characterize those, whatever function you were trying to design, you can actually see if these predicted completely de novo sequences have the ability to encode whatever function you were searching for. So as I mentioned earlier, like we were looking to build systems that can toggle between two different structured states and respond to some trigger, right? And that's the basis of allosteric cooperativity. So we actually just expanded this basic mode for protein design to design two sequences, or sorry, I should say two structures at once. So we take our initial backbone and we perturb it a bit to generate a groove into which you can slot in a synthetic peptide. So you have this pink state shown above and this blue state shown at the bottom, and you can go between those two. And then we ask our protein MPNN, our sequence design method, to identify a sequence that can simultaneously satisfy both structures. You can think of it as like the joint probability of amino acids across all the positions in that fold. And then we ask AlphaFold, does this thing actually fold into two different structures when you fold it in the absence and presence of this red peptide that is the trigger? And that's how we basically can generate multi-state, two-state switching systems. And then the second part is actually experimentally characterizing them. So what we showed here is actually after characterizing a whole range of these possible switches, or putative switches, like we showed that several of them actually do fold into the target structures when we solve those structures by empirical methods like X-ray crystallography. Here's one example of a hinged protein that we designed where it can go from this X state on the left to this Y state on the right. And the Y state is sort of this kinked open structure that exposes a groove into which this red peptide can fit. And we can drive the system thermodynamically from one state to another. And there's like a lot of validation and research into that. So that's the first part of the problem, right? Is to generate kind of the initial allosteric core that can bind to something and undergo a change. But that by itself doesn't result in allosteric cooperativity. You have to actually couple it to something else, right? So we have our allosteric core here. It can go from this X state to the Y state driven by the peptide. But what we want to do is couple it to some other function. And the other function is, in our case, because we're trying to get cooperativity, assembly into higher order complexes. So what we do is to exploit like another set of dimers or complex forming proteins in the lab, shown in green and orange here. And basically just attach those interfaces rigidly onto that switching core. So the way this basically works is to take your hinge, attach a green piece onto the N-terminal end and an orange piece onto the C-terminal end. And you can do this combinatorially over hundreds of possible examples of green and orange. And what that results in is the chimeric protein shown on the right here. It has three interfaces: a green interface that interacts with an orange protein, an orange interface that interacts with a green protein. So that's the basis of its self-assembly into higher order structures. And then lastly, it still inherits from its parent, like the ability to conformationally switch between two states via peptide binding in this case. So we have this three interface protein that can toggle between these two different states. And I think if it's not totally obvious why this would assemble into higher order complexes, maybe it'll be a little clearer in the subsequent slide. But basically, we can generate, using those same approaches I described earlier, we can generate enormous libraries of possible fusions where the angle and distance between the interfaces can be modulated. So in the vast majority of these fusion proteins, their fate is to ultimately just self-assemble endlessly. As their green ends attach onto the next orange end of its neighboring protein, it'll just spiral out into endless helical fibers, and that's not what we want, right? We want two closed symmetric structures, and these fibers are just a recipe for aggregation. So what we do is generate this library, throw out everything that doesn't close, and we identify the small subset of these fusions computationally that can actually close into these ring structures. So this is the simplest type of closed architecture that we can develop using these methods, these protein rings, basically. I'm showing one example here of a protein ring that goes from this squarish assembly on the left, it's made up of 4 subunits, to this pentameric assembly shown on the right, driven by binding to this small peptide. So if our hypothesis is correct, like it should be the case that this system actually has cooperativity because it can sample only these two specific oligomeric architectures. Yeah, so there's another problem with this whole schema of going from one ring state to another ring state. One possibility is that the ground state ring before you add in the peptide is just way too stable, in which case it will never be broken apart and assemble into something else when you add in the peptide. Another problem is that even if, I should also point out here that like basically um, uh, more negative energy implies higher stability. So that's why a deeper energy well means a more stable state that you can't get out of. So in the middle here is another situation where you go from where the triangle state, like so one of the states is actually less stable, then the ground state is less stable than the peptide bound state. But the problem is that you have to go over this huge energetic barrier to get from one state to the other, where you have to open up and do something unfavorable. So while this system will switch, the problem is that its kinetics will be extremely slow. So we have to find a way to make that energetic barrier low and also make the ground state not overly stable. And the way we basically did this, and this is the result of a bunch of empirical optimization over many rounds of design and testing. The way we ultimately managed to do this was to impose some kind of suboptimality on like the initial ground state ring. So over here, I'm showing an example of this like tetramer on the left, this four-piece square. It's actually slightly open. And thanks to the fact that it's slightly open, it's a little bit unfavorable, or the pieces have to flex to close the entire ring. And so what that makes is a sort of unhappy ring that almost wants to bind to a peptide and rapidly transition into this pentameric form, which it's more happy in because it has perfect closure. So using this approach, we actually were able to identify many possible pairings of different ring switches, which should obey the MWC model and have cooperativity. And I'm just showing the subset of them that I tested here. And even in this relatively modest set of rings, we're actually able to identify many examples of switchable dynamic assemblies. So on the left, I'm showing one example where you go from a trimeric ring system to a dimer when you add in this peptide. And if you look at it in size exclusion chromatography, where the x-axis basically represents retention volume, which corresponds to, so bigger stuff tends to elute earlier than smaller stuff. And you can see that there's a decrease in size consistent with the model that you observe when you add in the peptide. And at the bottom is a system that goes from 2 to 3 has an increase in size. So that's actually corresponds pretty well with our design model. And here's where mass spectrometry offers like a substantial improvement in the resolution and accuracy of mass determination. So you can hear the x-axis is the mass estimated from ratiometric contrast. And each of these vertical dashed lines represent the expected masses of different numbers of subunits in the complex. So 1, 2, 3, or 4. And you can see at the top here, this complex goes from being almost dab on the mass of a trimer to when you add in the peptide diminishes in size to being closer to the mass of a dimeric system. And similarly at the bottom, you can see it goes from being a dimer, a two subunit complex, to being a trimer. And in fact, this is further validated actually when we did electron microscopy on these systems and actually, imaged these particles on a grid at nanoscale resolution. So you can see here it goes from this triangular, almost recycled symbol looking thing to when you throw in the peptide, it becomes this nice little staple and goes from being something that's 2-piece to 3-piece over here. So the mass spectrometry and SEC and also electron microscopy all agree with each other pretty well in this case. And we show that it can extend to even higher order symmetry. So you can go from a trimer to a tetramer, like a triangle to a square, or from a square to a pentamer or pentagon. And this is also confirmed with both SEC and mass spectrometry. Across a range of different concentrations, the switch still operates. So you might notice the little sleight of hand here, like why am I showing you all these examples of like oligomers that or assemblies that change in the number of subunits they involve, when all of the examples I showed earlier involve a retention in the total number of pieces. So for example, hemoglobin remains a tetramer in both states. It doesn't break open and become a pentamer. To address this, we also try to see if we can have systems that retain their overall number of subunits, but change their shape. And here's an example where there's a ring made up of two subunits that when you add in the peptide, it goes from being this narrow slit to being this wider hole. And we can actually interrogate like the binding of this peptide using SEC if we just tag it with and without a GFP tag. So that adds an additional mass that you can use to distinguish in both SEC and mass spectrometry. And it'll become especially important for mass spectrometry when it comes to actually measuring cooperativity. So you can see here, like when you add in the peptide, there's essentially no shift in size, which is consistent with the fact that there's not much change in the radius. But when you add in peptide tag with GFP, there's actually a huge change in retention volume. And you can also see the preservation of oligomeric state with and without peptide in mass spectrometry as well. So we also showed that we can that these assemblies are accurate within single angstrom, a few angstrom off away from their design model. So we saw these structures with cryo-EM. This is one of the designs that went from being a trimer to being a tetramer. And if you compare the design model to the validated cryo-EM structure, they're almost perfectly superimposable, which reflects the accuracy or geometric accuracy of the design of the fusions between the hinges and the interfaces that mediate assembly. And this is particularly striking when you look at it at the level of a monomer, where they're basically indistinguishable when you overlay them. So that shows that our design methods are actually fairly structurally accurate. Okay, so now we're to the moment of truth here, right? So the whole point of this whole long arc of building these kinds of assemblies to see is, can we actually rationally design this really complicated feature, cooperativity? And so if you're wondering, like, what is a system, if you compare a system that's not cooperative to one that is, what you would expect in a non-cooperative system is that all of the binding events to the complex are completely independent. So as you raise the concentration of the peptide, what you see is just a binomial distribution of ring states, where there's species that are bound to 0, 1, 2, 3, or 4 peptides, rather than just 0 or all of them being bound. Whereas a cooperative system, you have an all or nothing response shown at the bottom here, where you go from nothing being bound to the peptide to everything being bound to the peptide. So this is actually very hard to measure biophysically by most methods. I think most methods are kind of an indirect estimates of cooperativity, but mass spectrometry actually offers a pretty unique opportunity to measure this with a lot of accuracy. So what we need to do is be able to distinguish between all of these different partially bound ring species, right? So the one way to do that is to simply take the peptide and then add on like some kind of mass tag. In this case, 30 kilodaltons from a GFP that you can just genetically fuse to the peptide and then express it in E. coli and purify it. Over here, I'm just showing like basically these are the 5 species that we want to be able to distinguish. We also wanted to generate a negative control. So if the MWC model is true, like basically any system that can sample just 2 different specific symmetrical ring states should be cooperative. But the corollary of that is that if you don't have those 2 ring states, then it's just a binomial distribution, non-cooperative binder. And it turns out we can generate that negative control just by installing cysteines at a particular location on the hinge and then freezing it in just one of the states. So the blue state I'm showing here. And this should be just a static ring that just binds to peptides without any change in conformation. And that should be non-cooperative. So what we did was to measure both of these systems across a range of possible peptide concentrations while keeping the total ring concentration constant. And what you can see here is these dashed lines indicate a trimeric ring shown in pink over there. And then different partially bound species, the masses of different partially bound species. So a tetrameric ring bound to 0, 1, 2, 3, or 4 peptides. And what we see here is actually kind of a beautiful transition in mass spectrometry from just all the rings being completely unbound to being transitioning cleanly to species that are fully bound. So you see that all or nothing exchange between trimer and fully bound tetramer. And if you compare this to our negative control, what you see is the static ring, it just has this ugly smear of different states as it transitions from unbound to partially bound and then fully bound rings. So I think the two-state design approach actually is the kind of an elegant validation of this model from 50, 60 years ago, and actually is an effective way to design systems that are all or nothing. So you can we can efficiently design cooperativity into these systems. And this is also true of the ring that I showed earlier that retains its size, the closest analog to the hemoglobin molecule I was showing earlier, where it goes from nothing bound to this dimeric ring, where nothing is bound to the peptide, to everything being bound with marginal occupancy of the intermediate states as we raise the peptide concentration. And then the next thing we did was to actually measure, or actually quantitate the cooperativity of these systems using mass spectrometry. So we did multiple measurements, or 3 measurements at each peptide concentration, and estimated the occupancy of the unbound form and the bound form, and basically showed that across a range of ligand concentrations, you actually do see this S-shaped curve corresponding to a switch-like response, similar to what I showed you earlier with hemoglobin. And by comparison, the best fit data, the best fit curve to this data, assuming no cooperativity, is shown in the dashed line here. And you can see that the empirical data is a substantially better fit to the cooperative model than the non-cooperative model for both of the rings that I showed. And we can quantify the S-shapedness or the sigmoidalness or the steepness of this transition using this parameter shown here as n, which is called the Hill coefficient. So Hill coefficient of 1 means no cooperativity. Any value higher than that does mean cooperativity. And what's striking is that if you take the Hill coefficient of hemoglobin, it's 2.5, whereas the systems we built actually exceed rival or even surpass this canonical example of hemoglobin in terms of its cooperativity. Okay, so we're done with the cooperativity part, and the next 2 parts will be a bit shorter, but also quite interesting from a more therapeutic perspective, rather than my nerdy interest in the biophysics of cooperativity and allostery. So this is work with a graduate student in the Baker lab, Abbas, who I mentor. Basically, we wanted to try and see if we can build cages that can theoretically enclose something. And then when they encounter this peptide or whatever effector or trigger, they break apart and release some payload. So part of this is to actually just build such switchable cage-like structures. So the way we did this was to take this trimeric protein up here, shown in purple, and then attach arms that contain these hinges onto each of the individual subunits within that complex. So then we have this trimeric flaw molecular design here, and then what we do is take that and self-dock it against itself via these weak interfaces. And this is all done computationally, I should clarify. And so there's weak interfaces with limited surface area of contact, and the idea is that when we add in the peptide, the hinges should bend in such a way that the overall architecture is broken and then basically falls apart. And we actually engineered 2 different kinds of systems here. So here's like a hexamer made up of 6 subunits. You bind to the peptide, and it becomes a trimer. Bottom here is like a decamer made up of 10 subunits. Bind to the peptide, becomes a pentamer. So it like splits up in half. And both size exclusion chromatography and mass spectrometry actually show that there is a shift in the mass and retention volume of these systems when you add in the peptide. And this, by the way, both these methods help us screen through many other examples where the cages ended up being static, for example. And this is actually also confirmed in negative stain EM. So where you basically throw in the peptide, and you can see that it goes from being this cage-like assembly at the top here to being these disarticulated floating trimers and pentamers. as revealed by electron microscopy. So the final thing I wanted to talk about is sensor design. So one of the main things in sensor design is to not only just be able to bind to your whatever target of interest, but and undergo a conformational change, but also connect that to something that can be read out on a macroscopic level. And one approach we tried to deploy to do this is to make allosterically inducible dimers. So every other system I talked about was breaking apart an assembly or going between different assembly states. So here we want to go from being monomers to being assembling it to something larger, and then the assembly of that is connected to some kind of signal. Here is a system that goes between these two different states. There's a pink state and then a blue state. So in the pink state, this thing has a hard time assembling with itself. It's the same category of rings made up of hinges connected to protein interaction modules. So in the pink state, it has a hard time self-assembling because it clashes with itself. But when the peptide binds, it kind of relaxes and opens up, and now is able to engage in productive interactions with other copies of itself. So that's like kind of an inducible dimer system when you add in the peptide trigger. So mass photometry also shows this, so it goes from being essentially a monomeric protein, C1, to when you add in the peptide, it becomes this dimeric assembly, basically. And this is a contingent on the concentration of the peptide. So how do we actually, so this is just like a system that can assemble conditionally, but it's not actually connected to any signal. So how do we connect it to a signal? And there's several ways actually. So one way is to just hook it up to a split luciferase system. So where the one copy of the one gene is expressed tagged with half of a luciferase protein. The other copy is tagged with a different half of a luciferase protein, or a different chunk of it rather. It's actually a smaller peptide. And when you express them separately, there's no light signal. And when you express them together, there's still no light signal or very little signal as a result of the luciferase assembling. And you also have to add in substrate that is converted into luminescent product. But when you throw in the peptide, there's actually a dramatic increase in luminescent signal consistent with conditional assembly into dimers. So you can directly connect binding to this peptide to a light-based signal. And it's a similar thing with fluorescence or a FRET-based assay. So basically here, you attach these two different fluorescent probes onto your proteins, mix them together, and titrate in peptide, which is shown on the x-axis here. And as you titrate in peptide, the fluorescent interaction between these two probes actually produces this FRET-based signal. So there's two different ways that you can actually couple this to either luminescence or fluorescent signal, both of which are readily readable in conventional plate readers. So the final thing I'm currently working on is extending this beyond just building systems that can change the oligomeric state, but actually directly turning an enzyme on, which is like one of the most canonical examples of allosteric regulation, where you have your peptide or some metabolite, a small molecule, it flies in, binds to your enzyme, and then makes it now process some substrate or reaction that can be easily read out. So it's sort of like a more direct extension of what I was showing earlier. So I've actually now had some success in basically adding in this red peptide here and making this enzyme move a little bit when it binds, and that actually registers as a 4 to 5-fold change in its activity as it goes from an inactive bad state where the catalytic residues aren't in the appropriate location to one where all the pieces twist and change and then reveal this cavity that can do the reaction. So this is something pretty promising, could be a new generation of, if we can generalize it to lots of different peptides, especially ones of biomedical importance, that could actually be a pretty important sensing platform. And that's what the future work is that I'm going to work on will be concerned with. So yeah, I mean, so to conclude, like the kind of the structural and geometric accuracy afforded by modern protein design tools allow us to access a huge range of allosteric functions that are previously not accessible to protein design whatsoever. And thanks to the accuracy of sequence design and backbone design, there's all kinds of work, sensing, triggerable delivery devices that hopefully we can build over the coming years. Because these kinds of smart medicines are like going to be a major factor in new kinds of therapeutics. And another thing from a more biophysical perspective is that actually you can use this rigid body fusion of different switchable components to do quite a wide range of different kinds of allosteric applications. And rather than trying to mimic all of the really subtle changes that you see in natural systems, maybe a more effective strategy for designing switches is to throw all of that aside and try to build them from scratch using de novo design, where we can readily rationalize and predict almost on a sheet of paper what kinds of changes will happen when you add in your effector. And with that, I'd like to thank a whole bunch of people, and especially my collaborators in the Baker Lab and the Washington Research Foundation for funding this research. Thank you.